So I'm going to do. Okay, so this is session 3. And, so like I said, we're just going to go through. Last time we did bands, which you remember on Friday. And so what we're going to do is we're going to go through what I call the lines. So let me just click here. Let me do this again. Okay, so remember what I was talking about again. I just like to reiterate that imaging and anatomy are not the same thing. It's imaging plus anatomy. And OCT is our favorite imaging modality. And remember that OCT is not a retinal biopsy. It's, again, a second-order interpretation of light and reflectance patterns based on differing optical densities and optical properties. And, normally it's like anatomy goes to the function and imaging, but the way we do it in ophthalmology is you see the image, then you learn the anatomy, and then you learn the functional aspect of it. And that's kind of the way we're going to be going through the retina. So just remember, you have to understand imaging. It's not just pathology. You have to remember the reflectance patterns, the dark light kind of contrast that we were talking about before. So again, here's your OCT, and then here's the pathology specimen. And histologically it's one way, but it doesn't necessarily mean that the imaging is going to always reflect exactly the histology. Again, here we're looking at our OCT. That's what we're trying to kind of remember we're going to try to figure out that we can memorize, but like understandably so. And so here we are again. And I remember I told you, we look at the foveal depression first, then we make sure that the vitreous, which we know is black, is evident. We see that in this case. Last time we talked about the bands. Today, we're going to talk about the lines, which I call lines. That's not a formal description of them, but you understand what I mean. So that's today. Now, remember, again, this is a reminder that we talked about on Friday. Things that are light have densely packed substances. Our kind of analogy, or our structural, analog of that was that they were axons. They were banging off of a bundle of axons or plexiform layers. And then reminder too is that things that are dark are much more like vitreous. They have loosely packed cells and a ton of fluid, which tends to be the nuclei or the cellular layers of the retina. So that's what it was for bands. So what we're going to do again is we're just going to point, but this time I'm going to point to lines, and you're going to tell me if something is light or dark. And again, we can call them bands, but because they're like part of the linear parts of the outer retina, I'm just calling the whole thing a bunch of lines. So if I point to that one, is that light or dark?
Light.
Good. So that's light. How about that one?
Dark.
Good. So that's dark. How about that guy?
Light.
Good. And then how about, again, that guy is light. I don't know why I have that repeated. How about that guy?
Dark.
Good. So that layer there, that kind of heterogeneous layer is dark. How about that?
Light.
Yes, light. Exactly. And then underneath it is also...
It's also light. Yeah.
Correct. So we just went through it. We're just going to do it again. I like to repeat myself. So what is that? Light or dark?
Light.
Okay. And then what is that one?
Dark.
And then this band here?
All of it is light.
Good. Light and light. I'm just showing that this is one of those instances where you have contiguous light layers, as we are going to be nomenclature wise. This is different. Remember, we're not in bands right now, but that's why. So I'm just showing you that you can have two light lines against each other just for this particular case. And then how about that line right there?
Light.
Good. So that's light as well. Okay. So I just wanted to show you that's one of the lines. Okay. Okay. So for the lines, just remember they are different from bands in the sense that generally speaking, we're talking now about individual cells. We're not just saying like, oh, it's an admixture of a bunch of nuclei, or it's a mixture of like, axonal, whatever. It's usually either going to be for lines, structurally a single cell, just to get the kind of mental framework going. And they're usually, all these cells are vertically oriented, at least as we're going to describe it right now. And they're normally packed tightly with neighbors in some layers, in which case you're going to get a light thing, because when you get something that's tightly packed, as I said before, they're tightly packed by our looking, but light can always find a way through the little spots and bang off of them. So what tightly packed just means is a ton of stuff that's right smack dab next to each other that light can bang off of. So packed tightly with neighbors in some layers. And then in other respects, so for other interpretations of a light layer, it's because the internal contents are super packed together. So again, this packing idea, if you keep it in your head, that's why something is light versus something being dark, in which case, if we take the cellular view, something is packed loosely with its neighbors in some layers, or it's like the cells are kind of diving away from each other. They're not actually connected together. Those are going to be dark if you can conceptualize it. And then the internal contents itself within, again, intracellularly now, those are packed loosely and floating around. Those are also going to be dark because light will just pass through an area of that particular cell inside of its cell because you're not having things for it to bang off of and reflect. Okay, so that's now the paradigm of light versus dark in this setting for lines. So the lines, what are they? We have the internal limiting membrane, which is light. We have the external limiting membrane, which is light. We have the myoid layer, which is dark. We have the ellipsoid layer, which is light. The outer segments of the photoreceptors, which are dark. The interdigitation layer, which is light. And then the RPE/Bruch's complex, which is light. That's just going over what we're going to go over. So this is from your book. You can see we're looking here and we can see that basically the retina is basically vertically oriented. This is the whole retina. This is not just one cell, right? We're just showing you that that's what we're looking at. But remember, so I'm just going to show you the first one. The internal limiting membrane is going to be kind of like an odd man out. He's up top. He's not. So we're just going to define him right now. It's the foot plates of the Mueller cells. That's what they traditionally say the internal limiting membrane is. But remember, the foot plates are connected to each other, and it's this foot of this Mueller cell. You can imagine him arranged upside down, where his head is down in the retina, but then up where the internal limiting membrane is. That's where the foot plates are, and they're connected. They're very close together, and so because of that, you're going to get a lot of reflectance patterns off of it because, again, packed closely together. So that's the foot plates of the Mueller cells, okay? And I'm just showing you, there's your Mueller cell. It's a photo that you have from your book.
Yes, yeah.
Okay, so that's the foot plates. Okay, so we're going to now show the rest of them. Now, mind you, notice we were here. This is the whole retina, the foot plates going all the way up. And if you can see to the right there, there's a ganglion cell and ILM. Okay, so we're really zoomed out of the retina. Now, when I move to describe the other layers, we're actually zooming in, and notice the outer plexiform layer is the top here. Okay, that's the top, the outer plexiform layer. The internal limiting membrane was the top in that other image. So we're already zoomed pretty far in. And where are we actually focusing? We're focusing these lines on this specific part of the photoreceptor. So remember, the lines aren't even the whole photoreceptor. Why? Because take a look, the outer plexiform layer and the outer nuclear layer pretty much are defined by this part of the photoreceptor. The photoreceptor is ginormous. And so that part of the photoreceptor actually makes up the bands and some lines. So it's literally some of the bands and the lines are made up of this huge photoreceptor complex. So just remember that. So it's like, it's not that like an individual cell can only be a bunch of lines and that's all it can constitute. Sometimes it can, parts of it can be the entire band. And in this case, the photoreceptor really stretches up. So we're focused on this spot right over here. And so I'm going to cut that away. You see how I've cut it away? And I'm like zooming in just on this part of the photoreceptor. So don't forget that it's not the whole photoreceptor. It's just this part of the photoreceptor that makes up most of the lines. And so from this particular zone, we're going to start figuring out what we're doing. So if we look at the external limiting membrane, which again is light, and you know it because you can see it in your head, I'm sure of it. It is made up of the tight junctions between photoreceptors and Mueller cells. So the opposite side of the Mueller cell, you can argue that it's its head, not its foot, is like upside down, and his head is actually connected to the body, if you will, of the photoreceptor in a tight junction. So the tight junction between the photoreceptor and the Mueller cell makes up the external limiting membrane. It's tight junctions, thus the light is tightly packed together. The word tight is in it. And so thus you get these reflectance pattern where you get a really nice hyper reflective line because the light is bouncing off of it, okay? And so you can see that's the schematic. Make sense?
Totally.
Good. So now we're going to go and move on. We're going to go to the myoid layer, which is right under it. Now, why is the myoid layer dark? We're going to tilt to one of our ideas about the intracellular contents. This is the spot where you get a heterogeneous mix of intracellular organelles, but it's not like just one. It's like a bunch of different ones. It's the ribosomes, it's the endoplasmic reticulum, it's the Golgi bodies, and very rarely there's a mitochondria floating around. But remember that it's dark. So that means these guys are not loosely packed together. They're kind of just swimming around. They're heterogeneous, but they're not like packed together. They're able to float around. The endoplasmic reticulum is like, this, it's like a highway that needs some space, and the Golgi's got like places to go. Like it's not actual, and the ribosomes are connected, but they're not packed together. They're actually dotting the landscape. So that's why this is dark. Now, this is and this is the area of it. Now, this is in contrast to the ellipsoid layer, which is right under it, and it's super bright. But why? Well, guess what? It's packed with a bunch of mitochondria, which look like ellipses. So if you look at a mitochondria, it looks like an ellipse, so therefore we call it the ellipsoid zone. We could have easily called it the mitochondrial zone, which would have made a little bit more sense, but we call it the ellipsoid zone for no particular reason, except to say that there were ellipsoid or intracellular organelles. And if you can imagine this part, and we're going to show right here, that's where the mitochondria are. But if you can imagine this entire thing, just letting you know, is the inner segment. This is the inner segment of the photoreceptor. Notice how it has if you want to consider the ELM part of it, the ELM, it then has, but the inner segment really has this myoid zone with these intracellular organelles that are very, very loosely packed. And then this dense kind of almost, and I like to think of it, if this was a glass, like a sedimentation of all the mitochondria that sit at the bottom of the inner segment. It kind of like sits down and it kind of all collects almost like garbage or crap or some kind of drink that you have where all the fruit collects at the bottom. This is like basically all of the mitochondria collect at the bottom. And so you get a hugely reflective line. And this is exactly why we always adjudicate the health of the photoreceptor by looking at the EZ or the ellipsoid zone, because the photoreceptor is a highly energetic cell and it needs mitochondria to function, because it takes something like I think it's like 1 trillion photons in a day. And if you have 1 trillion photons in a day and you constantly are generating an electrical signal for every photon, then you're going to need a lot of energy. And guess what? You have a ton of energy packed in the ellipsoid zone, and that's a marker for it. Does this make sense?
Yeah, and this is when you're talking about the inner segment, like so when we talk about the inner segment, outer segment of photoreceptors, you're saying all three of these make up what's known as the inner segment.
Well, I will say that when you're talking about, so you can argue that the external limiting membrane is sort of a dividing line, or like it's like saying like, is it in or out? I don't know. It's kind of like ELM is where we start, and then that's where the inner segments begin. And then the inner segment is actually comprised of, OCT wise, the myoid and the ellipsoid.
Perfect.
Okay. Now, so we're good with that. Now we're going to go to the outer segments of the photoreceptor. So notice something. The inner segment does not have just one line. The inner segment, I just told you, has a dark and a light line because of the intracellular contents. But then we move down a layer, or outer a little, and all of a sudden you get the whole outer segment gets its own layer. It's called the outer segments of the photoreceptor. Just live with that, okay? It's just the way it works. And why are they dark? And so I always say, remember, this is where you get cones and rods. And remember one thing, the rod, so there are two reasons of how you remember this is dark. The first is that, remember, a cone, if it's going to be sitting next to a rod, it already is going to cone away from each other. And you can see here, almost like it's fading away. So there's a space here for light to just pass through and not reflect. That's number one. So it's not as densely packed as its brethren up top, like the ELM. There's no tight junctions, obviously, because this is actually where the discs are shed. But then the second thing that I just want to explain, remember, if there is light and you see a white layer, then guess what? A white layer means that there was reflectance. But remember, if something absorbs light, it looks black, just like a black hole. A black hole absorbs light, and so thus it's black. Well, guess what? In this case, what do photoreceptors do? Where does the light get absorbed? The photon travels all the way down here and gets actually absorbed here, if it's gonna be absorbed by that particular photoreceptor, and then an electrical signal is transmitted, but no reflectance happens if they're working correctly. So there's two ways of thinking about it. There's a physiologic way, the photon is being absorbed in this space, and then there's your kind of geometric way where you say they're not densely packed together, so they're going to be dark. They're not going to be as light as definitely not as light as the ellipsoid, which has just the sediment of all this powerhouse intracellular organelles all collecting right in that same spot and aligned basically next to each other. Does this make sense?
Yeah, totally.
Good. Right, so I show you that. Okay, so remember, I just want to clarify, inner segments have multiple lines, outer segment doesn't, okay? Then we get to the interdigitation layer. So the interdigitation layer is down here, and it's basically where the RPEs, I don't want to use this word, so I'm going to use first the words apical processes. That's what they call them, apical processes. But I actually call them fimbriae, right? Because they're like fingers coming out, and they're almost like the seaweed where you interdigitate, and by definition, they're trying to fill the gaps, because what are they dealing with? They're actually dealing with the shed discs and recycling them. So they got to be as close as possible to the tips of these. And so if they're going to be like that, you're basically going to get interdigitation, and thus of the outer segments, and through the apical processes. So they're super packed, especially as compared to this guy where you got 2 reasons for it to be dark. So that's why you get pretty big light line, because you get this interdigitation effect, and thus there's a tighter packing of the actual structures. Does that make sense?
Yeah, totally.
Okay, so that's interdigitation. And then finally, RPE-Bruch's complex, which is light. Why is this true? Because these are tight junctions between the RPE. I will also just say that there is something called Verhoef's membrane. Don't get me started on why I have to mention that. It's because you see this line, because that's where there's like, you can see a slight dark line between the interdigitation zone and the tight junctions, because relatively speaking, that line, it's like less packed together of the RPE cells than the tight junction itself. So you get a dark light band, but you can see, don't worry about it. I'm just pointing it out. If you ever hear Verhoef's membrane, you'll be like, oh, on electron microscopy, you can determine that there's this line of the photoreceptors, because remember, I mean, of the RPE cells, because remember the RPE by definition is polar. What I mean by that is not the electrical thing, electrical argument. It's polar in the sense that it has apical processes and a basal layer, so it has directionality to it. And so you're going to get organelles just like the inner segments, kind of in one spot and not in another. That's, just remember that. So that just so that you understand it. That's why there's an apical process. Maybe that'll help you remember there's apical processes. But the other reason why I want to mention this is, yes, they're tightly packed, but the tight junctions also function as the second blood-retina barrier. So in other words, the first blood-retina barrier, and the blood-retina barrier also is the blood-brain barrier. The first one is the actual endothelial cells that line the retinal vessels, the ones that course through the inner retina. Those endothelial cells, they actually are the blood-retina barrier to, you know, the blood-brain barrier or the blood-retina barrier. But the second blood-retina barrier is actually this right here, the RPE tight junctions between each other, because where does the outer retina get their blood supply? From the choroid.
Choroid, yeah.
Diffusion through the choroid, you know, the watershed zone, etc. So there's a watershed zone within the retina, and just remember that thus you have to have basically 2 blood-retina barriers or blood-brain barriers. The first is the actual endothelial cells around the retinal vasculature. The second is going to be right here. It's this RPE tight junctions with each other that also, by definition, because they're so tightly packed, are going to be a light layer. Does this make sense?
Yeah, this is great.
Good. So multiple layer. Oh, and by the way, we call it RPE-Bruch's complex. That's how I want you to say it. Because again, we cannot on OCT distinguish the different layers of Bruch's membrane and RPE. We can only do that pathologically. So here it's the RPE-Bruch's complex. Make sense?
Yes.
Okay, there you go. It's down there. Okay, so we're looking at the holistic picture here. And we're going to say, okay, well, those are your bands. Remember this, we don't forget that there's the rest of the retina, but here's your bands: outer nuclear layer, outer plexiform layer. We don't have to care about this right now. We'll get into it at a later date, but that's your bands. Now we're going to go to the lines. Here's the inner segments with the ELM. Well, you have your ELM, your inner segments, then you have your outer segments, which is one layer, and then the interdigitation zone down below. And then you've got your RPE Bruch's complex. So, you know, if we go just to show again, right, here we go. This is where, I don't know why I'm doing this, but I'm just doing it for you. Here's external lipid membrane, light. Here's the 2 inner segments, dark and light, myoid. Here's the outer segments of the photoreceptor, which gets its own layer again, and then the interdigitation layer, light, and then you get your RPE Bruch's complex, light. Okay, so here we go. I'm going to point, and you're going to tell me what is the layer, and then I will tell you, I may ask you a follow-up question or I may not, okay? So here we go. We're gonna point right there. What's that layer?
So we've got the external limiting membrane.
Okay, and what is that made of?
It's between the photoreceptors and the neural cells.
Sorry, Nitin, you're actually going in and out.
Sorry. Is that better? It's the
Yeah,
tight
it's better.
junction.
It's better now.
Yeah, but tight junction between the photoreceptors and Mueller cells.
And the Mueller cells, you got it. Okay. What about that? That's the line. It's not the
Yep.
NFL.
Not the neurofibrillary. Yeah, that's the ILM, the
Okay.
foot
And
plate.
what is that of?
The foot plate of the Mueller cell.
You got it. How about that layer?
So that first dark line is the myoid layer.
And what is that made of?
It's like a mix of organelles, so the ribosomes
Yep,
and
got it.
endoplasmic reticulum. Yeah,
Perfect. How about that really bright layer?
ellipsoid layer.
And why is it bright?
Because it is the powerhouse of the retina packed with mitochondria.
You got it. How about that layer?
And below that is the outer segment of the photoreceptors.
Yep. And why is it dark?
It's filled with cones and rods. And so two reasons. There's, as you were saying, the cones kind of are shaped structurally in a way that allows for more passage of light, but then also they're photoreceptors. So they don't reflect back as much light because they're absorbing them.
Correct. So yeah. So the passage of light also means they're not reflecting back. So because it's going straight through. And then if it does make contact, it actually gets dark because it's being absorbed. Now, I just want to point out one thing. Notice how there's this like heterogeneity. There's a little bit of like reflectance here. That's partly because that's when they start to get involved with the interdigitation zone. So you can really see where it's just the kind of you could almost divide these two, right? And what Dr. Spade and some of these other people would say is that this kind of like schmutzy kind of area is the cone outer segment tips. They'll call it the cost line. I didn't write that down, but it makes sense to you now, right? Why would it be a little bit more kind of like splotchy here with a little bit more hyperreflectivity? Because the outer segment tips are actually starting to interdigitate. And so it's like right before the interdigitation zone, but probably right contiguous with it, there's like a smear in the line. Does that make sense?
Yeah.
And it kind of makes sense now, like you get it now when you look at an OCT, you're going to be able to say, no, I get why it looks the way it does. At least I have a framework, you know? So, okay, going specifically, what is that next line?
Moving to the interdigitation layer.
Right. And so what is that layer?
These are so the fimbriae or apical processes
Right.
that are connecting with the photoreceptors.
Apical processes of what? What is the apical processes of? Which cell?
Of the Mueller cell? No.
What is, so the Mueller cell's up top, right?
Up top, yeah.
Right. So here's your Mueller cell. Its feet are here and it comes all the way down and its head is here. So the Mueller cell is nowhere here. This is the RPE, right? So the RPE creates its fingers, right? So the apical processes of the RPE are interdigitating with your outer segments of the photoreceptors. Make sense?
Yep. Mm-hmm.
So there you go. And then what is that layer? What do we call it?
And we're calling it RPE/Brooks complex.
Oh, correct. And why is it bright?
It is very, very tight junctions because it's it's a blood retina barrier.
Yeah, so exactly. So there are tight junctions. That is a pathologic designation, or I suppose an electron micrographic designation. There are tight junctions between the RPEs cells. So that's a legitimate, you could point to it and there's a protein that connects them. The same thing up at the Mueller cells on the ELM between the Mueller cells and the photoreceptors. But this tight junction complex, this connection between two RPE cells, it's important and unique because it does function as the second or the outer blood retina barrier. Make sense?
Yes.
Good. So, right. So we're going to do this again. Okay. I'm going to point and you're going to do the same exact thing. Okay. Here we go.
Great. So I think we're pointing to the myoid later there, which is the.
Everyone does that. And I always laugh. I go reassess.
Oh, outer. No, it's the, sorry, it's the outer segment.
Yeah.
Photoreceptors.
I don't know what it is. It's literally, I have to tell you, there is like there's like 90% of the time, that's what people say, the myoid layer. I think that's what they like, oh, I got that dark one that I didn't know what it was. So they say myoid layer, but you know what this is. You just told me it's the outer segments of the photoreceptors. You got it. Okay. How about that?
And then that is the external limiting membrane.
You got it. Tell me what that's made of.
It's the tight junctions between the photoreceptors and the Mueller cells.
You got it. What is that? That's not, yeah, go ahead.
Yeah, internal limiting membrane, which we talked about is the footplate of the Mueller cell.
You got it. What's that one?
And then there we're talking about the think we're below. So we're at the RPE Brooks complex.
Yep. Tell me what's special about it.
Yep. It's the blood retina barrier. It's composed of tight junctions.
Right. And it's the outer blood retina barrier. What's the inner one?
It's the actual retinal vasculature.
The endothelium around the retinal vasculature. You got it. Right. So you're not wrong. I'm just trying to, like they may be specific on your whatever, your OCAPs or whoever, but you're absolutely right. That there is a sheath around it, so to speak, that prevents it from leaching outside of, let's say, diabetes or something like that, right? Okay, what's that one, your favorite?
My favorite, myeloid layer, which is the mix of intracellular organelles.
Yep, and you name one.
Ribosomes, Golgi.
Perfect. How about that guy?
Okay, so here we have the interdigitation layer, which are the apical processes, specifically of the RPE.
Yep, and what are they interdigitating with?
With the cones and rods in the above layer.
And what do we call, like, so you're saying cones and rods. Remember, the entire photoreceptor is called a cone, right? You're talking about the outer segment, right? So just remember that.
They interdigitate with the outer segment. Yeah.
Yes. So it's interdigitating with the cone outer segment or the rod outer segment, whatever, what have you. You got it. Okay. How about that one?
The photoreceptor layer.
Well, that's
sorry, the ellipsoid layer.
I was like, you are technically correct, but you're absolutely right. What is the ellipsoid layer made of?
Packed with mitochondria.
You got it. Perfect. Okay, you know me. Final time.
Yeah, let's do it.
Now you're just gonna tell me what the layers are.
Okay. So starting with the ELM, the external limiting membrane, and the ellipsoid layer, and the internal limiting membrane, and the outer segment of the photoreceptors, the interdigitation layer, the myeloid layer, the RPE Brooks complex.
You got it, man. That's it. You did really well. Is that helpful, right?
It's good. Yeah, no, it's nice to revisit this and go through specifically, the bands and then the lines and understand really actually the reason for their reflectivity. It's one thing to memorize them, but it's another.
Yeah, no, I will tell you that now we will build on all this stuff and we go layer by layer. Pretty soon, as I've said, and I've said this before, you will, once you understand why the layers, like what the connection is, it becomes not, I won't say trivial, but it almost becomes like, oh, right, like I can actually remember like what that is because I know how it works. I actually really like the build from the building blocks. It takes a while. So it'll take, like I said, it takes like 12 lectures, but it gets you back to saying I'm understanding it. So anyway, so tomorrow we'll do one, I think 7, if that's okay.
Yeah, 7's great.
Okay, good. And then to get some rest, all right?
All right, thank you so much, Dr. Diva.
No problem.
Yeah, take care.
Okay, bye.
Yeah.